So this is Alex's website. It's nano.physics.illinois.edu. And if you navigate, there's a tutorials tab you can click on to go to /tutorials here. And there you find, a bunch of tutorials, some very old, some newer, most of them in need of some amount of updating, but you can see a lot of them have to, from the images here, a lot of them have to do with these sort of systems that are not purely biological, but maybe they include some solid state nanopores or, DNA, but not DNA as a biological construct, but rather as something that we either want to try to read information from using a nanopore, or something that we want to build a device out of at the nanoscale. So the tutorial that I want to go over is on the second page, I think, here. And it's this practical guide to DNA origami simulations using Nandy. Now, it's a very, I would say this is it's a select group of people who actually wants to run simulations of DNA origami because they are very large and in general, and it's very expensive to simulate them. So not too many people are interested in simulating these, but we do have this tutorial on. I mentioned this mRDNA tutorial. That's this one here. It's not ideal for a workshop setting because it takes time to download and install the software packages that are required to run those simulations. But it is, to me, it's fun because and it also requires special hardware. It requires a GPU. Every only runs on the GPU, unfortunately. But it's it's cool because you get to see your system relax in almost real time. 20 minutes and it's done, and you have large scale transformations occurring in the systems. We also have this all-atom simulation of simulating membrane spanning DNA nanopore. That one's maybe actually like more of broader interest in the practical origami. But anyway, I was planning on going over the practical origamis tutorial. So click on this. Our website has a bug. You can ignore these. Okay, so then the tutorial comes with a pretty large archive. I guess it's not that large, it's only 100 megabytes. And then also the actual PDF document itself. So there's some introduction describing DNA origami. I think you probably got a bit of that from Alex's lecture this morning. It's already assumed if you're working with this tutorial that you have a file that describes the DNA origami nanostructure. That comes from the file that you'd have comes from the nano software package, which is it's a little CAD utility that mostly experimentalists use to try to design these DNA origami nanostructures. So I'll just load that up right here because we have it, and can get a bit of a feeling for what this software looks like. Basically, you have kind of two panels. The one on the left allows you to start immediately drawing something in like a kind of a cross section. So here I've created a 6-helix bundle, and then a DNA, and then if you click on each of these helices, you create strands. So right now we have so the 6 helices are represented sort of in a side view, completely unfolded in the right panel here. So we have helices 0 through 5, and just by clicking in the left panel, CAD Nano has already started drawing the strands, and you can create connections, and it gives you sort of hints about where you can create the connections. I'm not really needing to go into details about the process of designing origami, but maybe it's interesting for people to know that roughly this is how it's done by experimentalists. So this isn't a particularly nice origami structure, but fine. So that's what designing them looks like. They tend to get quite complicated in the hands of our experimental collaborators. Yeah, but if you feel like it, you can download this software. It's free. It's open source, and you can start designing origami sort of as you like. Okay, so then so once you have a file from CAD Nano, we have an online service that you can use, the EnergyMD web server, which does some relaxation of the structure using an all-atom model, but with no solvents and a ton of restraints. Amazingly, it works pretty well, actually. I think that's where we just kind of get lucky that it works. So oops. Let me unpack the archive and tutorials. And this, of course, can be done with the graphical tools, but for me, it's more comfortable to work with the command line, but it doesn't matter, actually. Okay, so our tutorial files, like this. So it's broken into, the tutorial is broken into 4 steps. And step 1 just has the single file hextube.json, which is this CAD Nano design file. So here. Path. And then we go to this web service. We upload our design file. We select the lattice type. That's unfortunately not encoded in the design file. I mean, it can be guessed at, but our service doesn't do that. But we know it's a honeycomb lattice because it's, yeah, you have when you construct the origami, you can have a honeycomb lattice or a square lattice. I mean, others are actually possible, but those are the 2 that are widely used. Yeah, with a 6 helix bundle, you have sort of a hexagon of DNA. That's honeycomb lattice. We'll just accept the default options and then call it hextube. Create this simulation. So this is just generating, this is actually not running the simulation. This is just generating the all the Nandy files that you need in order to run this energy and simulation. So it takes this design file, automatically converts it to PDB, it generates a PSF, and also the elastic network file, and it also writes a Nandy configuration file. So we get a little archive, which you can now download, and we can, sorry, let me make this larger. You can follow along. So I'm just moving that that file that the web service just generated to the step 1 directory, and then we'll unpack it. Again, you can do this with the GUI. There's no problem with that. Okay, so the archive contains a directory called hextube, and in that there's a number of files. Never mind the chicken wire files, but we have a PSF, a PDB, a Nandy file, an extra bonds file, Charm 36 parameters with the ion corrections that I'm sure Alec talked about in his lecture, and a README file that describes the contents here. And we can already open this PSF and PDB in DMD, just inspect it, make sure everything looks okay. And okay, I'm sorry, I'm running over FastX. I will open a session on another machine that is not having a visualization error. So this is my laptop here is not very powerful, so I'm running with a remote desktop. What is this? Okay. So we can immediately load the DNA nanostructure into DMD, inspect it, see that yes, it is the hex tube that we supplied, but you can see that it's very idealized at this point. We have these long crossover bonds that connect the backbone of a strand on one duplex to the backbone of the same strand, and really on the other duplex. And you can color it by, for example, chain. Set name anyway, to see that where each strand is located. So the typical thing, you'll see that there's one strand that's in all of the helices that's blue, and that's typical of origami because you normally have a piece of viral DNA that you're trying to fold up with the staple strands. So and then yeah, the viral DNA is folded up by these shorter staple strands that are all kinds of colors in this representation. Okay, let me pull now the tutorial back up. Okay, so now yeah, so so 3.1, step 3.1 introduced you to CAD Nano. The version I had doesn't have the nice visualization in the panel, in the third panel, but it is possible to install it as a plugin to the software Maya so that you get this 3D visualization. Yeah, and then I just brought you through the process of uploading the file to our web server, where it'll generate the PDB and PSF files. And then you download the results and you look at them to make sure that everything looks okay. And you can see these long bonds. So then the next step is to just run the MD simulation. I think that this computer already has things running on it. It's certainly slow enough that it seems like it. Yeah. But actually, GPU 1 is pretty open. So you might just have Nandy 2 alias, and then you can do make it large again. So you might just type Nandy 2 xtube.nandy. Well, what is it? Plus P16 or something. Up to 10. And that would run without a GPU. And oh, maybe this is running with a GPU, actually. So you run it with Nandy in the usual way. In my case, I hit a CUDA error, which is probably because of the unusual characteristics of the system. With some older versions of Nandy, I don't think I hit those errors. But if you do hit it, then just try running with the CPU. If you're simulating with one of these Origami nanostructures. They have unusual characteristics that because there's no solvent in the system, it may not work with a GPU. But because we don't have solvent, also the simulation is relatively fast to run. Yeah, so I'll just leave this running for now, and we'll just talk about the next steps. Yeah, so I guess the tutorial asks you to inspect the Nandy configuration file. I think that's fine, but a little boring maybe. I guess the one kind of unusual thing is that we use the extra bonds feature to create this elastic network of restraints. So basically, every atom in the bases of the DNA, if there's another atom within something like 6 angstroms of that atom, in the idealized structure, we've got between an extra bond that's added, and that prevents the DNA from falling apart, which normally if you don't have counterions, you would expect it to really repel itself because DNA is very highly negatively charged. But with these elastic restraints, it stays intact and it keeps its B form quite nicely. So then we run the simulation, and we can, I guess, start already visualizing what happens to the structure. And at the end of this, we'll have a dot coordinate file, and we can load that into DMD and save that coordinate file as a PD structure for subsequent solvated simulations. I guess I'm pretty quick here. So I guess I actually did this in the wrong spot. Technically, what I'm simulating right now should be step 2. The tutorial supplies all those files as well. So you can actually skip step 1 and go straight to step 2. So you don't have to upload the design to our server to still try things out, but it's easy to do. So yeah. So typically when people create these DNA origami objects, they fold them in fairly high salt conditions. Usually they include something like 10 millimolar magnesium chloride, in addition to maybe 200 to 500 millimolar sodium or potassium chloride. And magnesium has a valency of 2, so it tends to be attracted to a charged thing like DNA very strongly, more strongly than the monovalent sodium or potassium ions. So we generally have a significant amount of magnesium in the simulation in our origami simulation systems, because they should have a large amount to mimic the experimental conditions. Typically, we in our lab we've just neutralized. First, we neutralize the DNA origami using magnesium and not at all using the monovalent counterion. And in addition, we may add a small amount of magnesium to the solution. And normally, you add some mixture of ions. The total system is charge neutral. You're close in molality to the target that you want, but the thing that you kind of have to do is wait for ions to equilibrate around the DNA, and then you can actually look at, in a corner of your system far away from the DNA, you can look at what the ion concentration is there to get a sense of what the bulk ion concentration is for your system. I think that strategy is not limited to origami. I think in ND accumulations in general, that's a good way to think about and to report actually the ion concentration in your system. So I recommend, keeping that in the back of your head, even if you never ever want to simulate a DNA origami nanostructure. So the other thing that we spent a decent amount of time on, and this is actually came about a bit as a process of trial and error by Jejung, who's the first person in our lab to really start simulating these origami systems. So he found when he used the standard charm ion parameters, including magnesium, he found that his DNA kept getting stuck. The DNA molecules would stick to each other, and he thought that was incorrect because it's not. I mean, it's not reported that way in experiments. And he did a bit of searching and found, first of all, actually, he demonstrated that it wasn't correct by reproducing osmotic pressure data. So there's data that goes back, I don't know, 30 or more years, where the pressure in a system containing multiple molecules can be measured, and in an experiment by kind of counterbalancing that pressure with something, with a system that's partitioned, where you have an agent that's producing osmotic pressure, like PEG, for example. That data exists for DNA molecules in an array, so a bunch of parallel DNA molecules. There's experimental data that says, at what distance between the DNA molecules you would relating the distance between the DNA duplexes and the pressure in the system. So Jejung simulated that system, a bunch of DNA molecules surrounded by a cylindrical potential. And in simulations, you don't need this osmotic agent. You can actually just directly measure the forces that are applied by your restraints, and derive a pressure in that way. And when he did it, I mean, there was no pressure in his system because the DNA was all stuck together, mediated by the magnesium ions. As a result of that, Jejung decided that probably, first of all, magnesium should not make direct contact normally with the DNA phosphorus atoms. In experiments, I think he had a few lines of reasoning for that. It's described in his paper, which is on this topic. I'll just, I guess, bring up that paper real quick. It's from a few years back already. Paper. No, that's not the paper, sorry. This is the paper. The structure and intermolecular forces of DNA condensates. So not showing. The figures are not showing up. I just wanted to give you a sort of a visual guide. So this is actually after the correction was applied. Yeah, I think he doesn't have a figure here of before, unfortunately. But the DNA phosphorus, maybe there's a different paper that talks about the condensate with sorry, with plain magnesium. But anyway, in this paper, or a closely related one, Jason laid out some rationale for why he would expect the magnesium ions to be hexahydrates actually, and not forming direct contacts with the DNA phosphorus atoms. And he found that when he forced the DNA to be hexahydrates using some extra bonds restraints between specific water molecules and the magnesium. And also when he applied a small NB fixed correction to those water molecules, he was able to reproduce the osmotic pressure really nicely. So this panel down here shows the experimental data in black and the NB simulation data in red. So that's here is sodium only, but in the right it's with magnesium. Okay, sorry, that was a little bit long-winded, and apologize for that. But I just wanted to give a proper background. So we always have magnesium ions in these systems, and in our lab, we generally force those magnesium ions to be magnesium hexahydrates. It might be that if you had a good polarizable force field at your disposal, this would be unnecessary. But as of now, with tip 3P water, this is merged as the best approach for modeling DNA origami nanostructures. Okay. So after the energy MD simulation, which was check there is. We still have a little ways to go. You can see it's relaxed already quite a bit. So after this energy MD simulation is complete, you can see that the crossovers are now you don't have these long bonds anymore, but you also do see some distortion in the helix. The helix on the right has lost some base stacking interactions here, which is not great, but that would be resolved by simulating further. So I'll probably just start using this structure for the next step. Just pretending that it's simulated for long enough with this network of elastic restraints. Slide. So I guess to that end, to write a PDB out. There's a script that does this for you, but I mean, it's simple enough to do. So I just create an atom selection. I don't know if you guys have experienced the TK console before. If you all have experienced that. But under extensions, TK console, you can type in, there's a number of, so it supports TCL, which is a scripting language, a general purpose scripting language. And it supports a bunch of DMD specific commands like atom select, which we use to create an atom selection. So some slice of the atoms that are in the molecule that's currently loaded. And once we have that atom selection, we can write it out to a file. We'll write it out to, we'll just call it hex2 min.pdb. And then so I will copy that file hex2 min over to step 2. I guess I won't because it already exists there. So there's a better example of this file already packaged with the tutorial, which I forgot about. Okay, so back to step 3 where we're setting up the system. We have a script, a TCL script that adds these magnesium hexahydrates for us. And copying and pasting, but I'll just type it. Yeah. And so I think here we're saying we want to add 516 magnesium ions, which is half the number of nucleotides that we have, and we'll call the resulting file hex2_mghh, which stands for magnesium hexahydrate. So this runs VMD as in the back, not in the background, but just in the line, and executes the commands in the script with the arguments that were provided here. So we basically specify the PSF, the PB, the output file name, and the number of lines to add. And it takes a little while for this to run, not terribly long, but a little bit of time. So then once this is finished, then the next step is just to solvate the system. Often with origami, the structures are so large that they break the solvate plugin in DMD. So I can't recall if that's true for this structure, but if it is, yeah, let's just have a look at what's in this script. Sorry, I forgot. Okay, yeah, we just do use the solvate plugin, and the autoanionize plugin of DMD to add the solvent and the ions to the system. So this is a simple enough script. And it would have to be customized if you're using a different structure. This is one of the places where you would change things so that you have your system's dimensions and not these arbitrary ones that are for the hexagonal tube here. I guess before we proceed, I should visualize this hex tube structure. We can load both the GSF and PDB, and we can see that we have all of these surrounding the DNA. We now have a bunch of identical magnesium hexahydrate groups, like the ones shown here. So each of these has a pink magnesium ion in the center, and it's surrounded by 6 water molecules, and there is we'll create an extra bonds file that will put restraints between the oxygen atom of those 6 water molecules and our magnesium ion, so that it has to stay solvated during the entire simulation. The problem is, or one of the problems we found, once a magnesium ion loses its solvation shell and makes direct contact with the phosphorus atom, it never regains its solvation shell on the time scale of simulation. And that's consistent actually with experiments, I think. I think the time scale for those kinds of transitions for magnesium is on the order of microseconds. Yeah, so that's maybe expected, but it is problematic if you're running a 100 nanosecond simulation. But the main thing is that if you allow the magnesium ions to make direct contact with the phosphorus atoms, it bridges between 2 phosphates, and it causes artificially strong attraction between the DNA duplexes, which are in complete qualitative disagreement with the experiments. So by using a small ND fix correction and by enforcing hexahydrates, we're able to get quantitative agreement actually with the experiments. So that's what we do. Okay, so this is again what the structure looks like with the magnesium. I'll close this. And then the next step is to create the magnesium hexahydrate extra bonds. We have a Perl script that ships with the tutorial. It's mghhextrabonds.pl. I personally don't like Perl. Jaejun wrote this script, but some people might. So I don't think there's any reason though that you would need to modify it. Like some of the other scripts, this one is fully automated. Sorry. Okay, so now we can look inside this file and see that we've got restraints that are 10 kcal/mol per angstrom squared, and they're 1.94 angstroms. These are bonds that are 1.94 angstroms long. So so just at the idealized length for the magnesium hexahydrate. So then we would need to include the extra bonds in the Nandy configuration file. They are already there. So for example, in this minimization script, I'm gonna make this a little bit smaller. Okay. Yeah. So so it's fairly standard Nandy configuration file. We have a few, there's a proc here for getting the first time step of the simulation from whatever XSC file was provided. I don't know if this actually gets used. Yeah, cell basis vectors, parameter files, 8, 10, 12 switching cutoff and pairless distance. The femtosecond time step. PME, this PME is a little too generous, actually. It should be 1.0. I think that was probably, I don't know who did that, actually. And the Langevin damping here is a little bit high. For equilibration, it's probably fine. For the very early equilibration, it's probably fine to have a PME grid spacing of 1.5. But in general, you don't want to go up above 1.1 or 1.2, in my experience. That's treading into territory where things might not be accurate enough. Yeah, so then here we highlight 2 extra bonds files. The first is the Elastic Network that was generated before, and then by the online server, and then here we also have the extra bonds file that we've just created. And all this simulation is meant to do is minimize the structure. So the strategy here is to did I solvate the system? Hold on. Sorry, I might have skipped a step. Yeah, I think I did. I did skip that step. Yeah. I apologize. I lost my spot and I almost skipped the step where we solvate the system. That won't change anything in our extra bonds file because the atomic indices for the magnesium hexahydrates will stay the same. The solvate plugin and the auto-ionize plugin both just add things. They add water and ions respectively to the end of the PE and GSF. So they won't mess with our existing indices. So we can keep our magnesium magnesium hexahydrate extra bonds even though we've done this step out of order. Yeah. Actually, while I wait for this to finish up, does anybody have any questions at this time? No questions. Great. So it looks like we're all done with our adding ions and water. We can now visualize our system. It's hextube MGHH underscore WI for water and ions. .PSF and TV. And just like with the membrane systems, you always want to make sure that there's plenty of solvent, around your solute particle. So here, the nicest way to do this is to visualize the system using QuickServe, for example, and a transparent representation, a glass bubble. Radius just a little bit bigger. Click with something like van der Waals, just to get a sense of the scale. Now remember, the diameter of DNA is something like 20 angstrom or 2 nanometer. So, so you can tell immediately just visualizing that we have at least 2 nanometer on all sides of the DNA construct. You could actually, this box to my eye, it looks a little generous, but during equilibration, sometimes things contract a bit. So it's okay, I think. It should not be in class. Something like this. So, so actually, at this point, our system is fully assembled. It's actually easier to set up these simulations than even a simple protein system, because you don't have to worry about disulfide bonds or histidine protonation states or any of that. But yeah, so I think actually DNA origami systems are straightforward to set up in terms of their molecular composition. Where it can be difficult is that they can be quite large. Like this is a very, by experimentalist standards, this is a small DNA origami nanostructure. I mean, like almost trivial. I don't think they would ever, most experimental labs would never synthesize this or assemble it. But already we're at 430,000 atoms to represent this system. So you can imagine if you do go to a larger system, in practice, you're looking at running millions of atoms. If it's like a real structure that you have an experimental collaborator who's working with, it'll be in the millions of atoms. So then again, the next step is to equilibrate the structure. It's not so different from equilibrating a protein system, for example. We start by having these restraints, and we want to remove those restraints gradually rather than all at once. The protocol we came up with was to run for about 5 nanoseconds first with a spring constant of 1/2 kcal per mole per angstrom squared for each bond in that elastic network, and then 0.1, then 0.01, and then finally remove the restraints altogether. And the Nandy configuration files for doing that are already a part of the tutorial. They're provided, but you can inspect them, and you can also look at the differences between these. For example, with the diff command. Issues with my read line. That I'm writing. Oh yeah, no, that's fine. Oh, I didn't mean to do that. Those are actually the extra bonds, not the Nandy configuration files. So the only real differences are a variable that gets changed for keeping track of what's used as input for the initial conditions, and then what we call the thing finally after the simulation, and then the contents of the extra bonds file for the elastic network. Yeah. So then. Yeah. So I think I won't actually run the simulations because you'd have to wait some time for them to finish. But you could. normally I would run these simulations, and if you were working the tutorial, you would run those simulations. If anything went wrong, you know, just email, ask for help. Yeah. And then at the end, when you're done applying the restraints, you would run that simulation as a production run, as long as you care to. And 5 nanoseconds, by the way, is a pretty, that's around the time scale where you see the ions, you know, they'll equilibrate quite, you know, pretty thoroughly in about 5 nanoseconds. So if you start out with a really, you know, not so good ion distribution after 5 nanoseconds or so, it's most of the way better. I mean, you probably would need to simulate 20 nanoseconds or so to really have it be fully converged. And again, it probably depends on the size of the system. You know, in general, a larger DNA origami structure, for example, it'll take longer for an ion at the edge of the system to propagate inwards. But 5 nanoseconds is plenty of time for the ions to relax. For the DNA, we see for other structures that are a bit larger than this one, we see that it relaxes, you know, more on a 100 or 200 nanosecond time scale. And some of them would fluctuate, you know, indefinitely. I mean, the more flexible structures would just would fluctuate probably on the microseconds and longer time scale. I think even the DNA duplex really, you know, goes through quite a bit of fluctuation on the microsecond time scale. But yeah, that's been our experience, though, that if you take an idealized nanostructure, it'll converge towards its equilibrium structure on maybe 200 nanosecond time scale. That's very rough, though, because the specifics, I mean, it'll depend on what the actual system is that's being simulated. For this tube, I think it doesn't take so long. So the last part of the tutorial walks you through some simple analysis that you can do in VMD, and also just I think mainly using VMD. So the first thing that you know the tutorial has you do is it has you look at the size of the system. So in figure 6 down here, it shows that the system box equilibrates within the first, I don't know, half nanosecond or quarter nanosecond or something like that. And then, and I'm sure you've done this in other tutorials this week, the second step in the analysis is to plot the RMSD. And what we see is that after an initial very large jump upwards, there's a slow increase as we release the restraints, where the root mean square deviation increases modestly. But that, I guess, yeah, that's sort of an indication of how quickly your system equilibrates. Although I don't, you know, from this data, you couldn't say, oh, this is equilibrated. This is not been simulated long enough to say definitively whether or not the RMSD is still increasing. And then the third step has you run a TCL script that counts the number of broken base pairs. So we find that for this structure, the base pairs, there's only a few base pairs that are actually broken during the simulation, which is good. Most of them should be intact. And the script measures the broken base pairs using distances. I think it's a really simple, it's just about distances between the atoms involved in the Watson-Crick base pair. And then to visualize the ions, we look at the total charge within 2 nanometers of the DNA. And that's also done with a TCL script. Feel free to have a look at those scripts. If you have questions about how they work, of course, you can reach out to us, and you can you can maybe adapt those for your own systems. Yeah. So in this case, yeah, the charge is very quickly equilibrated, it seems, and it's nearly zero the entire time, actually. So that's actually reassuring. So this is the total charge of ions and DNA. So what we see is that for the ion concentration here, it's the origami nanostructure is basically charge neutral within a 2 nanometer shell. Yeah, the last step of analysis has you draw the origami structure as so-called chicken wire representation. So that's where instead of drawing every atom, you can just draw the central line of the center line of the bases, and you can draw bonds at the crossovers where those strands cross from one duplex to the adjacent duplex. Yeah, like shown here, and that's a helpful representation for seeing how the structure relaxes. And that's basically the contents of this tutorial. I don't know if anybody has a question about it. If you do, please just let me know, and I'll be happy to answer. Okay. So let's see. So we still have some time. Let's see. So maybe I'll actually I'll quickly just skim this tutorial, the multi-resolution tutorial. I guess I mean it lets us the techniques described in this tutorial allow us to do studies like this one here, where we ran a simulation of a DNA origami plate and being captured in a pipette. And this plate was designed with fret dies and that were attached near this this double-stranded DNA leash that's used to capture the plate. And we were able to measure the distance between the dies and compute a fret signal, and make remarks about the functioning of this structure as a possible voltage sensor, which was the name of the project. So these are coarse grain simulations. They're much cheaper than all atom simulations, but yeah. So because they use a much simpler representation. Yeah, so I'll just quickly run through this tutorial. So first of all, the software is the big obstacle with performing this tutorial. You have to be able to have a machine with CUDA, a CUDA-enabled GPU, and I'm afraid it's only been tested on Linux, really. I mean, I've built ARBD before on Mac, but I actually haven't built it on Windows, and I'm not sure if it would work or not. It probably would through the Linux subsystem, but I haven't done it. Yeah, and besides needing ARBD, it also requires the mRDNA package, which is non-trivial to set up. It's not terribly difficult, but it helps to have familiarity with installing Python packages. We have a GitLab where, I guess I'll just, oops. We have a GitLab page where the mRDNA package is located, and that page contains a script that in the README that can be used to install mRDNA using Conda. Yeah, that's what I recommend. If you want to try this out. So the tutorial is pretty simple. The mRDNA package, the PUM package comes with some command line utilities that you can use to just take your design file, like that hex2.json file that we were looking at earlier, and basically automatically, just with a simple command like this from your terminal, it will create this simulation directory and create all the files needed to run the coarse grain simulation. So performing, first a very coarse simulation to get the rough sort of structure equilibrated, and then at high resolution to deal with the orientation of the duplex throughout the structure. And then finally, we mapped an atomistic representation, which you can solvate and simulate as shown in our origami tutorial that we just went over. So it's a really simple utility to use. The difficult part is, I guess, having these JSON files is creating those. And it exposes some simple options. So you can control how long you want the simulation to run, how long you want the coarse simulation to run, how long you want the finer simulation to run, how frequently you want to output the results. It has a bunch of other options available as well, but it's just an example. Yeah, the tutorial then, suggests some scripts you could write to do some simple analysis. I mean, you can also just use the plugin in DMD. There's nothing wrong with them. They're perfect. But for sometimes you want to customize your analysis. So that's why we show how you can do it here using the TCL scripting language. Similar, yeah. So I think this tutorial has you computing things like ARMSD, ARMSF, pretty standard. It also shows you how to, if you did have like a donor acceptor pair for fret measurements, and you knew where those were located in the design. The tutorial shows you how you would find the distance between them in order to estimate what the fret would be, what the fret efficiency would be. Then the next step shows you how to run Nandy. So this is this file that's produced, this Nandy configuration file. It's very similar to the one that we got from our server, but instead of starting from a perfectly idealized configuration where all the helices are straight, we ran coarse grain simulations to relax the DNA. But otherwise, it's very similar. It also uses an elastic network of restraints to keep the DNA intact, even though we simulate it in the absence of an electrolyte. Yeah. So then, yeah. So then, so for a straight DNA origami nanostructure like the hexagonal tube up here, not really why you would run these coarse grain simulation steps. They don't add, they don't really relax things that much, But there are a lot of structures which, if you just that are designed, for example, to have a curvature. So this is done by adding DNA on one side of the structure and removing it from the other side of the structure. And in the design tool that experimentalists are using, everything just is straight. I mean, they keep track of how curved things should be by using tools like this one. And there are others. There are alternative tools as well that they use. But it's not, there's no cue from the CAD Nano tool to say, actually, here's what the 3D shape should be for this DNA origami structure. So if you have a design that has a programmed curve like this, then it really becomes, if you want to run an all-atom Nandy simulation, you don't want to spend all of this time waiting for the all-atom model to relax from an initially straight conformation to a curved one. And in fact, you might have problems starting with this initially straight conformation because the bases would be on the side with extra DNA inserted. Those bases might actually be clashing with each other. So for systems like this, it really is necessary to have a tool like MRDNA. And again, it's simple to use. It's just the same as before. You can also simulate some nanostructures that are a little more complicated. This, for example, is showing a slider system that came from Carlos Castro's lab in Ohio State University. So there you have these two subunits and the 6-helix bundle in the middle. The blue subunit at the top of this is fixed with respect to the 6-helix bundle, but the green one can move up and down. And experimentally, you can lay these down on a grid and take cryo images, and then carefully measure the distance between the two pieces of the two bundles of DNA, and come up with an extension. And we can do something similar, just computationally. And I think that the tutorial guides you through that process as well. I actually don't remember. Yeah. So and we find that our coarse grain model isn't quite good enough to give perfect agreement, but it's okay. There are also things that are not origami that people are doing. There's a whole class of nanostructures that are wireframe meshes. People simulate things like these boxes with the idea that maybe you could put a protein that performs some enzymatic activity and maybe somehow deliver this into a cell. And then if it's in the right cell that has the correct mRNA key, you could open the box using a strand displacement reaction, something like that, selectively delivering its cargo. That's the idea. I don't know. I mean, no one's managed to make this principle, I think, work yet, but that's sort of the dream. And you can model these kinds of structures and try to make improvements to designs using this tool actually. Others as well. There are a few other tools. Yeah. So up to this point in the tutorial, it's basically showing you different kinds of systems that you can run all pretty much the same way with this simple command. Just mRDNA, and then here's my system. However, sometimes your model you might need to do something a little more complicated, and in that case, this is the mRNA package is written in the Python module, and you have all of its internals available to you to set up and manipulate these kinds of systems. So the last portions of this chapter, first of all, talk about some of the details about what's really kind of under the hood of the model, and then it provides you with a Jupyter notebook that you can walk through and learn about some of the lower level representations interactively. So I'll just I won't actually run the examples, but I'll bring it up. So if you go to TBGL, instead of tools, you go to tutorials. So gitlab.ngr.illinois.tbgl/tutorials. It's linked to in the tutorial. You can go to the multi-resolution DNA nanostructures tutorial, which is what we have here. Well, I guess it's actually a part of, it would have been part of the archive if I downloaded it. But we pull up this IPython notebook, and yeah, actually, this isn't rendering properly. Maybe I should actually bring it up. It's fairly large, actually, this tutorial. Sorry, I should have done this ahead of time. Okay. eventually this will work. Okay. So so the tutorial just shows you basically, how you can, read a file in from, and create one of these MRDNA models just using this read model command, and it shows you how to use different parsers that we have available. And then it encourages you to inspect, what's actually inside of that model. So our models are composed of something we call segments, which are really like duplexes, but they could also be some single-strand DNA. And it's basically just a representation that goes from like a contour length of 0 to a contour length of 1. And then we use splines to describe how those segments are curved through space, and also how their orientation, is what the orientation of the duplex is. And it's possible, it's fine and to create your models like this. It is also possible to develop them sort of building from the ground up. Like you can create a segment just with this command. So 1 second, sorry. Okay. Well, another second, sorry. Sorry about that. Okay. So you can create segments manually with, to create DNA of whatever length you want, and it'll arrange it from, the origin to, some, by default, just some spot in the z-axis here. That's the length, the appropriate length for the DNA. And then you can also create single-stranded segments, and then you can connect them together using a variety of methods. That's basically the idea behind the tutorial here. And then if you want, you can take something like this, and you can simulate it and visualize the results, and change the resolution all through this scripting interface. Most of the time you don't want to do this because it's tedious, but it's nice to be able to, because occasionally you something when you're modeling something that just doesn't fit with a cookie cutter automated script. So the tutorial has you, first after teaching you these sort of low-level things, it challenges you to try to create a DNA mini circle, which has, relevance to in biology, of course, because of histones. And it's actually maybe not trivial to set up a system like this, but with the package, it's not too difficult. The next step of the tutorial shows you more kind of concrete use cases that we drew from our research. Oh, actually, sorry. No, this is still about low-level scripting. My apologies. The very last part is so yeah, this is still about like how you can control the splines and all of that. It's fine. And then the last part draws from our research and maybe shows some more interesting systems. I guess the second half of that was from a real research example. Yeah, and then the last one shows like an idiosyncrasy for how you would have to model something. In the CAD design for this sensing DNA origami plate nanostructure. In the CAD design, the leash, this really long double-strand DNA, was included using something called an insertion, which just says, hey, add 700 base pairs of DNA here. And that's fine, but the crossover next to this leash basically shouldn't be modeled as a regular crossover, and it becomes necessary to use the scripting interface to fix how the model is formed. Yeah, it's a bit detailed. I kind of want to spare you all the pain of going through it, but besides showing you how to fix that kind of modeling error, this step shows you how to apply a potential that's derived from a continuum model, say in a software package like COMSOL. Specifically, it shows you how to take data from COMSOL and apply it as a potential in an ARVD simulation, so that you can get a sense of what the how an origami plate on top of a nanopore like this, how it gets deformed by the electric field. Yeah. So I think that's all I really want to say about this tutorial. I mean, of course, we could work through the details, but I think that would be probably painful for the few who remain. Yeah. So I guess at this point, does anybody have any questions? Yeah, if anybody does end up working through these tutorials, if you have any questions about it, you can always contact me for actually for either of the two tutorials I showed. I'd probably be the person to contact. I guess the last thing I'll mention is that we have, and these are probably of more general interest, we have some tutorials about how you might visualize these DNA origami nanostructures. They just sort of showcase a bunch of different techniques for visualizing these things. Some of it's specific to origami, but some of it is not. So you might find some tips for how to make some nice images from this tutorial. I think everybody likes to make pretty pictures for their papers, so that your work gets showcased well. So yeah, basically shows how different representations look and all of that. We also have a related tutorial that's not about origami, but it's a little bit older and might not use all the latest features that Tachyon, the renderer, has to offer, which it's a really, I mean, I like Tachyon a lot. It's fast and it's, you know, it looks pretty good. We also have this, you know, this tutorial on rendering with Tachyon. Quite old now, but it does still cover, I think, 90% of what you can do with Tachyon. I mean, for example, this is an example of how it's old. The draw styles that are highlighted are Van der Waals, New Cartoon, MSMS, and Surf. This is predating the Quick Surf representation, and I almost would never use either of these other two surface representations anymore now that we have Quick Surf, because it performs pretty much as well, and it's so much faster, especially if you're making an animation of a trajectory. In that case, Quick Surf is the way to go. But it gives you an overview of sort of the materials that were in BMD at the time when this was written, and you know, it gives you suggestions about how to color things. You know, I think, you know, this is less relevant now than it was when this was written. But you know, sometimes people still print things in black and white. Yeah. So those are the tutorials that I really wanted to highlight. Yeah. If there's no questions, I don't know. Yeah. What now?
Kind of quiet.
Yeah.
But that was great, and this has been recorded, so we can post this in YouTube, and you can use that.
Hi, future YouTube viewers. Give us likes and thumbs ups or whatever they have on YouTube.
So yeah, if there are no more questions, I propose we close for today and for this workshop. I'm gonna send an email around for all the participants to fill an evaluation form, which has a few questions about things if you enjoy the workshop, how relevant it was, and also some open suggestions for you to contribute. So please fill that form. And yeah, I think it's thanks for being here, those of you who stayed until late, and thanks, Chris, for your great talk, and hope to see you guys sometime soon.